is with great pleasure that I'm introducing Marie-Laure Charpignon. She's a PhD student at the Institute for the Systems and Society, IDSS, at Harvard MIT Department of Health and Health Sciences and Technology. Sorry. Now she has multiple advisors, I think 2 or 3. I know Leo Seli and Maimouna Mayoum there, but I don't know if there is a third one. Is there a third one?
Yes, my main advisor is Munzer Daley in EETS at MIT here. He's also the director of our institute, actually. Yeah.
Right. So, so Marie's interests include causal inference, agent-based modeling, applications to public health, and so on. So, I mean, I can go on for a long time here, but I can mention that she obtained a bachelor in engineering sciences from École Centrale Paris and a master in computational and mathematical engineering from Stanford. She's worked at Microsoft, and she had internship experiences at Goldman Sachs, I think the European Commission as well, and at INRIA, I think. And I know her because of the workshop so I started to get to know her work because of a workshop in 2021 at ICLR that was on AI for Social Good. And it was a very interesting series of lectures and activities. And today she's going to talk to us about causal inference in medical records, application to drug repurposing for dementia. So thanks a lot for your participation here, Marie.
Yeah, thank you for the invite, Alessandro and Kathleen. And I noticed that some of the posters are actually related to Alzheimer's disease and either like measurements of brain volume changes and how that can, facilitate, I guess, data mining for drug repurposing and or detection. So I would be really interested in, hearing about your research on that front as well. And yeah, I just wanted to mention also, although I've never been to Indiana, one of my good friends at IDSS, Yuan, is now an assistant professor in information systems. And so that's how, I got familiar with several of the departments at Purdue. And also we've been doing some research on excess mortality estimation during the pandemic with the Indiana Department of Public Health. And I noticed that also another of your students is working on the dynamics of the epidemic in Indiana. So that's another topic, I guess, that we have in common and that
you're
we
gonna
can
come
talk
visit
about later. Yeah, so today, I would like to discuss with you the topic of drug repurposing for unmet medical needs. And the focus will be on the case of antidiabetics for Alzheimer's disease and related dementias. It's a collaboration actually with Mass General Brigham and Harvard Medical School here in Boston, as well as with Imperial College in London. And this research is a joint work with Beng, who's a recent graduate from Imperial, now at University of Edinburgh. And our postdoc mentor, Bella, who's now an assistant professor in Israel in statistics. And we have been lucky to work with Colin McDermott, who's a data engineer at the hospital here in Boston, and to receive guidance from Dr. Mark Albers, Sudash Nadas, Deborah Blacker, and Joanna Tulaki at Imperial. And this manuscript was first posted as a preprint on medRxiv, and it should appear later this month in Nature Communications. And yeah, if you have any questions or suggestions, I would be really happy to hear your feedback, because we're currently extending this work to other drug classes beyond antidiabetics. We're looking at antihypertensives, but also at medications against rheumatoid arthritis. So during the Q&A, I would be really glad to discuss with you, and your recommendations would be much appreciated. So I'll try to motivate this work a little bit before we start jumping into the details of the statistics. So why are we looking at other applications for antidiabetic medications. And why are we focusing specifically on the case of Alzheimer's disease and related dementias? So let's take two perspectives, one being global and the other one being US-centric. So across countries worldwide, life expectancy has been increasing. It has moved from 46 years old in the 1950s to 73 in 2022. However, this increase is paralleled with a rise in the prevalence of aging-related diseases, and these include cardiovascular pathologies, but also neurodegenerative ones. And globally, there are about 47 million people who are affected by dementia to date. And this number is expected to double, if not triple, by 2050. Now, looking specifically at the US, the increase in Alzheimer's disease and related dementias is a major concern because it represents a huge burden for a system that's already, costly and ineffective. And to date, there is no approved and deployed disease-modifying therapy for AD that has no side effects. So maybe you've heard the big news yesterday. Pharmaceutical companies are investing in novel therapeutics, such as this antibody drug, lecanemab, by Biogen, but toxicity is often a challenge. So another idea consists in exploring retrospectively and/or prospectively the potential of drugs that are already approved by either the Food and Drug Administration or by the European Medicines Agency. But instead of using them for their on-label use, it means using them off-label. So which of the drugs that are already on the market to investigate first? And the question is, would certain compounds, if any, be more effective than others at slowing the development process of Alzheimer's disease? So, as you probably know, dementia is a multifactorial disease, so it's associated with several types of illness, and actually there are several subtypes of dementia, including vascular dementia, Lewy body. And among the diseases that are related to dementia, there's one of particular interest, which is diabetes. And diabetic conditions manifest in multiple ways, and they have cascade effects. So similar to renal failure, which is a complication of diabetes, the impairment of cognitive function is also a downstream consequence of diabetes that is actually induced by hyperglycemia. This fact makes type 2 diabetes the second risk factor after age for the development of dementia. And given the increase in the prevalence of diabetes in the US, there's great potential in studying the repositioning of antidiabetic drugs and in evaluating whether certain medications could have a differential effect and delay the onset of dementia. So if we consider this risk factor, type 2 diabetes, more closely and look at some metrics, we realize that the rate is already high, but it could reach up to 20% in certain areas of the US by 2030. And taking a historical perspective, this progression has actually been quite fast. In 2004, the diabetes epidemic was mostly affecting southern states such as Louisiana, Alabama, Mississippi. However, 15 years later, in 2019, the map looked quite different. And over 10% of the adult population, 20 and older, has a coded diagnosis of diabetes at the time, according to a CDC surveillance report. And you can find, updates of these reports on a regular basis. Although these estimates do not distinguish type 1 and type 2 diabetes, the latter accounts for 90 to 95% of cases. So, you can think that this is like the underlying estimate of interest here. And the reason why I insisted on, coded diagnosis is that actually about 13% of the US population, is uninsured. There are behavioral health practices, including routine care, checkup visits that vary from, one place to another. There are also issues related to the coding of chronic diseases by GPs. And finally, various centers in the US do not necessarily operate together. So there's progress being made on that front. By the end of 2022, actually, provider organizations must use EHRs certified according to the 2015 regulations to be compliant with the Promoting Interoperability Initiative by the Centers for Medicare and Medicaid Services. However, because of all of these factors, this map is actually a lower bound of the underlying disease prevalence in the country. And that's the reason why it might be more informative in parallel to look at the geographical distribution of correlated risk factors associated with type 2 diabetes, namely obesity and physical inactivity. And these maps highlight heterogeneity among different counties in the US, with prevalence rates as high as 40% in certain areas. So now that I think I've convinced you of the potential of studying anti-diabetic drugs, let's come back to the connection between dementia and type 2 diabetes. So as mentioned before, diabetic patients are at increased risk of developing dementia. And specifically, 20% or more of older adults with type 2 diabetes could suffer later from mild cognitive impairment, which can aggravate and then lead to a form of dementia, which makes people, it makes people at increased risk of being able to maintain a daily routine, and that also places additional constraints on their household and caregivers. And patients with MCI will develop Alzheimer's disease at an incidence of between 6 and 25%, which compared to the general population is large. In the general population, it's between 0.2 and 4%. So overall, type 2 diabetes, you can think of it as being associated with a 1.5 to 2.5-fold increase in the risk of dementia. So that really motivates us to, study the repurposing potential of these drugs. And among the mechanisms that are known to intervene in both diseases, one can think of inflammation, of oxidative stress, and also mitochondrial dysfunction. So Kathleen can correct me there, but so yeah, that's the part of the cell in the cytoplasm where respiration takes place. And that's one mechanism that is involved in both type 2 diabetes and in dementia. So these factors together are known to aggravate, two things. One is brain insulin resistance, and the other one is amyloid beta accumulation in brain lesions. And prolonged exposure to hyperglycemia and hyperinsulinemia, as well as high levels of this protein, amyloid beta, in the brain, can lead later to neurodegeneration and therefore to the deterioration of cognitive function. So there are valid reasons to first focus on antidiabetics as a candidate class that could be repurposed for Alzheimer's disease and related dementias. But there are many kinds of such medications. So which ones can be considered first? So we started by investigating, the hierarchy or the tree of recommendations defined every year by the American Diabetes Association, which includes first, second, third line monotherapy treatment for type 2 diabetes. But as you can imagine, these guidelines evolve over time, which makes the retrospective simulation of a clinical trial more challenging. So just to take a little bit of a historical perspective here, in the study, we looked at two historically first-line antidiabetics, metformin and the sulfonylurea drug class, which includes many agents. And the reason why I say historically is that nowadays sulfonylureas are generally recommended as a second-line treatment. So that's the reason why we had to be careful with comparing these two treatment arms. So why is it interesting and relevant to actually compare these two arms? So the rationale is that they do not have the same mechanism of action. While sulfonylureas mechanism is based on the stimulation of insulin secretion by pancreatic beta cells, which results then in lowered blood glucose, insulin secretion actually remains unchanged with metformin therapy. Instead, the drug acts by decreasing hepatic glucose production and intestinal absorption of glucose. So that's the main difference. That's really, if we find anything, one of the potential reasons for that difference would be the mechanism of action. Another reason why it's relevant to compare these two treatment arms is that sulfonylureas have been widely used for the treatment of type 2 diabetes for over 50 years, while metformin usage is more recent, and especially in the US, because for a while the FDA was investigating the drug due to side effects, especially among patients with chronic kidney disease. Another potential reason, which I started exploring, is the potential physician preferences for sulfonylureas over metformin. That could be because sulfonylureas are cheaper. That could also be because under that monotherapy, you're only required to take 1 pill a day instead of 3 pills. And so depending on patient adherence and behavioral health, certain physicians might prefer that drug for specific patient profiles. So these three reasons, make the case for comparing drug A, metformin, versus drug B, sulfonylureas. And as mentioned, unlike sulfonylureas, metformin doesn't cause hyperinsulinemia, which, as mentioned before, can lead to neurodegeneration. So this additional element suggests that metformin might have unique benefits also because of that feature, because of the mechanism of action. But one problem that we faced initially when designing this study is that metformin is a bit chaotic in the sense that its history in the US versus Europe differs. So in 1995, the FDA finally approved the drug for patients with type 2 diabetes, but with prescribing restrictions. And for a while, there have been safety trials. Although in Europe, the drug has been in use since the 1950s. So because we studied, patient populations in the UK and the US, we also had to keep this in mind. So now that I've exposed several, design considerations, let me briefly describe the study objective and the underlying data sets that we've been using. So the objective here is to test the hypothesis that metformin reduces the risk of dementia onset among type 2 diabetic patients. And for this, what we've done is emulating or simulating in silico a synthetic drug trial that compares metformin to the other drug class of interest, sulfonylureas, and we've done so in two distinct databases, one from the US and the other from the UK. And the rationale for doing so was that, there were different histories of drug prescriptions in these two countries, but also if we were to replicate the results in the UK, then we would have more confidence, about the veracity of that effect, in the sense that these two countries also correspond to very different settings. That includes differences in medical practice patterns, differences in data collection, in data missingness, in known and unknown sources of bias. So we would have more confidence in the potential generalizability of the drug's effect. Yeah, go ahead.
Yes, so I have a question here. You might have said it, but I might have missed it already.
Yeah, don't worry.
Yeah, it's about how to use the two data sets. So do you so as you mentioned, the history, everything is different there, right? So in practice, they have completely different characteristics. So how do you work on that? Like, do you do some sort of, so yeah, that's a question.
Yeah, no, that's a good question. I'm going to talk about that. So
Yeah.
for now, we've considered the two populations separately.
Right.
So we conduct one analysis in the US where we rebalance treatment arms.
Right.
And then we conduct the same analysis in the UK where we also rebalance treatment arms. But for a while, we've been wondering, what about trying to define some sort of default population, which we could pick to be, let's say, the metformin population in the US, but it could be like any other population. Or we could actually formulate the problem as like maybe there is a barycenter population, which is a fake population, but it's the one that's, minimizing the distance from any center, any arm to that barycenter. And we could just try to like generate a big trial. So this is what we are trying to do now, because we have Israel and Denmark as two additional centers. So the goal will be to use these federated learning approaches, although right now they only account for one stage. So they only, for example, consider, one outcome being time to death or time to dementia. While here we first have to rebalance, or we have to, include a first stage for example, in the instrumental variable scenario, the relationship between physician prescription preferences and the patient treatment. So that's why we're working on this now, because I guess your question is, instead of conducting like 10 simulations in each country, what about trying to find a procedure that will account for the differences in mortality rates, the differences in coding and reporting of dementia in the medical records, also differences in comorbidities, etc. So that's what we're trying to do right now, although this particular analysis was really one simulation, rebalancing arms, another simulation, rebalancing arms. But ideally, we would like to find either a default population or conducting sensitivity analysis to the choice of that default population, or like build a barycenter that would be, representative, assigning weights based on the population in each site. So these are the ideas we're currently working on, and trying to integrate like two stages. One being the first stage of the instrumental variable procedure or propensity score model, and the second one being, several outcome models, one for dementia, one for time to death, but you could imagine also time to cardiovascular disease or time to cancer.
Got it. Thank you.
Yeah, definitely. So we have two main databases. One is coming from the Mass General Brigham Medical Record System. So that covers a total of 8 health centers in 3 states in New England. So that includes also New Hampshire and Vermont centers. And really the core of the database includes patients who are followed in Boston at Mass General Hospital or at Brigham Women's Hospital. And you could think, mostly these are going to be patients in New England, but actually when, conducting some zip code level analysis to understand better the social determinants of health, I realized that we also had a significant number of patients coming from Pennsylvania and Florida, for example. And in the UK, the data set is much more diverse. So patients are coming from 13 regions, more than 700 practices across the country, although the enrollment of practices, changes because GPs decide whether or not they want to contribute data to CPRD. However, overall, the UK data set is much more representative of, a nationwide primary care network than our US data set is, because it mostly consists of tertiary care hospitals. So just to summarize, these are the research questions that we've been trying to address. The first one is, what's the effect and what's the causal effect of metformin on dementia onset among type 2 diabetic patients relative to this control set of drugs, sulfonylureas? in doing so, can we in parallel estimate the benefit of this drug towards longevity? We also wondered whether these effects were constant over time, because often in clinical studies, you'll see a hazard ratio, less so a risk over time, although this starts being the case with more and more clinical trial simulation results. And finally, are the results, independent of the choice of the statistical approach? And these questions could also be formulated in the general population, right? You may wonder about the benefits of this particular drug among diabetics as well as non-diabetic patients. You can imagine that this is, harder to simulate, although this is an aspect that we're also working on. There's a trialist at Columbia University that's investigating this drug among non-diabetic patients. So we're partnering with their group to actually predict what would happen among patients that are, similar to diabetic patients, although not, receiving treatment before enrollment to the trial, and although they don't have a coded diagnosis of diabetes. And finally, there's a part of the team that's working on in vitro systems pharmacology studies, where they essentially in culture expose human neural cells to different substances, and they try and measure which genes are differentially expressed under, different drug dosage scenarios. So let me describe the approach that we've taken to try and assess the effect of this drug on both longevity and time to dementia onset. So we emulated a target trial, or simulated, a trial in 2 cohorts. And we've mostly leveraged the methods formulated by Jamie Robbins and Miguel Hernan since 2016 at the School of Public Health at Harvard. So this is a visual representation of our simulated trial. We followed patients with type 2 diabetes from time T0, which is their treatment initiation, onwards. We selected patients aged 50 and more. And we've only selected patients who initiate on monotherapy, meaning that if they initiate on both drugs, then they're not eligible to enter our simulations, so we exclude them. The Vanderbilt criterion for, type 2 diabetes normally includes also a coded diagnosis of diabetes. However, here we were mostly interested in drug exposure. So we relaxed that condition, and we considered, only being exposed to one drug or the other, irrespective of whether there was a diagnosis in the medical record or not. Although most patients would have a coded diagnosis, actually. But you can always conduct sensitivity analysis to that. And actually, to ensure that we would, have sufficient drug exposure before measuring outcomes, we imposed that run-in or grace period of 1 year after treatment initiation. The role of this running period is also to ensure that we don't have reverse causation, because we wouldn't like, the outcome of dementia to be observed during that period and attributed to one drug or the other, because that seems very unlikely. So this running period had actually several, motivations. And we used the pre-period to assess the severity of diabetes at baseline via 2 different markers, one being BMI and the other one being baseline level of hemoglobin A1C. And so one of the advantages of observational studies as compared to randomized control trials is that we have a much longer follow-up time period. So when you think about the time between T0 and T1, that can be up to 30 years in medical records, while, for example, the trial that's being conducted currently at Columbia University is only going to be for 2 years. And most of the time, it's not going to go beyond 3.5 years or 4. So that makes it really difficult to investigate, long-term outcomes, such as time to death, time to dementia onset, without, having the benefit of following the patient for many years. So we had, though, to make some decisions regarding, at what time we would start collecting medical records. We considered in the US prescriptions from 2007 onwards, and there were two reasons for that. One, the guidelines formulated by the American Diabetes Association changed in 2005-2006 approximately. And two, the Mass General Brigham system switched to an electronic system in 2007, which led to better quality data. And so we would rather, limit the amount of missingness in this simulation. So that was the reason why we made that design choice, to help us mimic as close as possible the settings of a randomized control trial. And in the US, really, what makes this type of study possible is the large adoption of EHR. It's over 90% right now, and it increased by more than 9 times since 2008. So the digitization also of clinical notes to find, blood pressure measurements, hemoglobin A1C measurements that have not been entered in structured data but are only available in the text, also makes this type of study possible. And, my hope is that in Europe, including in my country, in France, things improve over the next few years, so that information exchange both within countries and across nations, across EU member countries are made possible. So we really have to be careful with, using the right set of inclusion and exclusion criteria when we try to simulate randomized control trials using observational data. And here, I think, taking the example of this contraindication for metformin, but not for sulfonylureas, namely chronic kidney disease, I hope I convinced you that it's not just about running simulations, it's also about being very careful about study design, so that any effect that we measure is actually a causal effect. So I wanted to give you a brief overview of the methods that we've been using. So we estimated the effect of the effect of the drug on both time to dementia onset and time to death without dementia. So these can be viewed as two competing events. For this, in terms of the statistical approaches, we have two main stages. The first stage consists in rebalancing the arms, because naturally there are differences that occur among the different patient populations. So we adjust for age, we adjust for sex, for comorbidities. I'll describe this in more detail later. And the second stage consists in weighted regressions. So using the weight for each patient, we will have rebalanced populations, and we build survival models for each outcome of interest in these rebalanced populations. And in order to test whether the assumption of a proportional hazard between drug A and drug B was valid, we also use non-parametric regressions where we don't make that assumption to test whether the results were similar or not. And as in any causal analysis, we needed to specify a priori which quantities, which metrics we would be interested in. And we chose to focus on two quantities. First, hazard ratios, because these are used by clinicians oftentimes to describe the benefits of one drug over the other. But also importantly, we looked at cumulative incidence functions that describe the risk of developing dementia, or the risk of dying by a certain point in time. The reason why they're so useful is that clinicians are decision makers that need to prescribe one drug or the other based on the risk or the probability that you as a patient will experience, encounter this disease by a certain point in time. And so it's much more useful to provide a risk-based perspective than to provide a single hazard ratio describing the benefits of one drug over the other. So I just want to spend like a few minutes talking about the benefits of using a competing event framework. So why not just looking at dementia onset as the outcome and forgetting about any other outcome of interest? So here you can think of the different paths that a patient can take from treatment initiation onwards. So a patient can follow path 1, which goes from treatment initiation to the development of Alzheimer's disease and related dementias. They can also follow path 2, die without any accumulation, for example, of amyloid beta in the brain. They can also follow path 1 and then develop Alzheimer's disease and then die, which is path 3, and we haven't really focused on that. The reason being, there's bias in reporting of Alzheimer's disease, first off, in the EHR, so that makes it tricky, given the social determinants of health, then to study time from Alzheimer's disease onset to death. But one could also get interested in the benefits of taking one treatment over the other on path 1 and then path 3. And so what are the benefits of using that competing event framework? So one, we avoid misestimating the probability of event occurrence because we're including in the survival function the fact that certain patients will die without having dementia onset. And second, it's more meaningful clinically because physicians can assess different risks at a given point in time. So they can balance when they decide to give this drug, the side effects, the fact that this drug can be beneficial with respect to a certain disease, but not with respect to the other. So there are trade-offs, and that really helps to consider multiple outcomes in competition. So talking about these two competing events, how does that articulate statistically? So we will have two distinct hazard models. These can, either relate the two treatment arms, like in a Cox proportional hazard model, where there would be a coefficient beta 1 that will apply in case the treatment is equal to 1, and it will not apply in case the treatment is 0. Or we could also build, in a non-parametric framework, a hazard model for each treatment arm and an outcome combination. So one for metformin and death, one for metformin and Alzheimer's disease, and similarly for the other drug. So that's how it articulates in practice. And just to show you, like, mathematically where that matters to make this choice of considering two competing events. So if you look at the probability of having the event Alzheimer's disease or the event death without dementia onset by a certain point in time t, this will be written as an integral, and that encapsulates the survival function. So this s sub t is the survival function by time s. And how do we define it? Well, we define it as not having dementia onset and not having died without dementia onset. So this survival function is actually the product, although that doesn't, represent any sort of like product of survival functions. It's a product that involves both hazards, the hazard of death without dementia onset and the hazard of dementia onset. So that's why it's so important to encapsulate the risk of death without dementia in the framework. Yeah, Alessandro.
So this was always under the hypothesis of Cox proportional hazard model. Is that correct?
So yes, this is under the hypothesis. Yeah. Otherwise, we would have actually 4 cumulative incidence functions, and that's what I will show next. When we do a non-parametric setting, we don't have this beta 1 and this beta 2.
Right.
So also here to simplify, I haven't written the dependencies with the covariates,
Right.
but in fact, in this exponential, if you want, you can also include, if you want to use a doubly robust approach, you can also include the contribution of age and sex and cardiovascular disease and all of the other covariates that we've adjusted for in the first stage in the propensity score model. So here, just to simplify, this is taking the assumption of a proportional hazard, hence the fact that we have these two cumulative incidence functions under treatment T that are related, but we can also make it unrelated. And secondly, we actually tested that, the importance of doing additional control in the outcome model for the same covariates, the same confounders that we already adjusted for in the propensity score model.
Yeah, got it. The other question related to this model is, so then you have three compartments, so they're not called them. Maybe it's not the correct word, compartments. So you have, this just triangle there. But in practice, you have a more complicated structure because, there might be other, there are many competing risks in general.
Yes.
Right. So it could even be that you just add an other compartment, I don't know what to call it, another something there,
Yeah.
which really makes things more complicated, but it might be useful in that in the analysis or something.
No, this is super interesting, and I really like your question. So, in fact, the package that we wrote for that, you can consider any number of outcomes. So you can consider, for example, dementia onset, cancer, and death without dementia onset without cancer. You can also consider cardiovascular disease because it's another risk factor due to the relationship with vascular dementia. And that's one of my next steps, actually. I want to figure out what kind of uncertainties it brings to consider, multiple arms. So if a physician has to decide, okay, this drug, this other drug, in the case of antihypertensives, it's much more complicated because you have calcium channel blockers, you have ACE inhibitors, you have ARBs. So their clinicians have to choose among different drug classes, and then for each drug class, some drugs are known to penetrate the blood-brain barrier, some are not. So it's really like they have the choice among 30 drugs, and also they're trying to mitigate the risk of all the diseases, right, at the same time. So that makes much more sense to me clinically to try and consider multiple outcomes, not just two, not just the disease of interest and death without that disease, right?
Right.
And especially that can help, understand, for example, why cancer is supposed to be protective against dementia onset.
Right.
Because here, what we do is that we consider cancer history as a covariate. So we assume that it's part of the characteristics of the patient that may actually trigger one treatment or the other, while also affecting, time to death without dementia and time to dementia, because it's supposed to be a protective attribute. But it would make more sense to include, cancer as another competing outcome rather than, solely including it in the set of covariates that pertain to the patient. So I completely agree that also in terms of uncertainty estimation, it would be nice to understand what the implications are going to be, because any probability of events by time t that we would provide to the clinician in a visual manner, we like it to come with a confidence interval, right? And so what if we have 10 competing events? How to read that graph that balances all the risks by a certain point in time for a given patient. So yeah, like this is a consideration that I have in mind, and the package is designed this way. Of course, the problem is always, I guess, estimation and also challenges of making sure that in the medical records you've incorporated all of the ways that cancer can be captured, for example.
Right.
Yeah.
Good. Thank you.
Yeah. So here, actually, I was just discussing, these different aspects. The fact that we would have, covariates included, etc. So let's spend some time talking about how we define the covariates that will enter the estimation framework. So because we're not solely interested in associations but in causation, we need to spend time thinking about sources of confounding. And these confounders, as we were just discussing, are factors that typically weakly influence the treatment but strongly affect the outcome. And because here we have competing outcomes, confounders would be those factors that influence either dementia onset or time to death, or both at the same time. And in our particular scenario, we can think of confounders as part of one of the three, classes: sociodemographic characteristics, social determinants of health, markers that are related to the underlying disease. So here, the severity of the diabetes at baseline, but also spatial-temporal factors. So we were discussing earlier about the changes in guidelines, both in the UK and in the US. We also talked in the UK about the geographical heterogeneity across the 700 practices. So the region of residence might play a role here, both in terms of affecting treatment decisions as well as, environmental exposures that could be related to the outcome. So there is a rationale for each factor here. Of course, age is the largest risk factor for dementia onset and death, so it makes sense to include it. Why does it make sense to include sex here? Well, it's unknown whether it affects diabetic treatment assignment, but it strongly relates to death outcome, and it also relates to dementia onset outcome. So, if it affects strongly the two outcomes and maybe just weakly the treatment, it also makes sense to capture it. Socioeconomic status is related to both outcomes as well. And BMI, hemoglobin A1C are, characterizing here the severity of type 2 diabetes at baseline. And the other risk factors are those that we just discussed. So hypertension, cardiovascular disease, and stroke are comorbidities that are associated with dementia. And in particular, you could think of vascular dementia as a subtype. And cancer is associated, obviously, with time to death. And here, for a patient to be characterized as having, history of cancer at baseline, what we did require is at least 2 instances of ICD-9-10 codes. The reason is sometimes history of cancer within the family will be captured. And also in prior work, the number of instances, has been used as a way to improve outcome ascertainment. So we tried to inspire there from the existing literature. And in the UK, it was additionally controlled for smoking status as well as COPD, which are both associated with time to death. And to adjust for geographical heterogeneity, mostly in the UK, in the US, we do so through zip code that helps us define socioeconomic status, but that's the only thing we do. In the UK, we did adjust for the 13 distinct regions in the propensity score model that is aimed to rebalance the treatment arms. And because the time span is much longer in the UK, going from 2001 to 2017, we add a, one hot encoding for each of the years of the first prescription. In the US, there were less concerns about this because we started the data collection process in 2007. And in fact, when looking at, the number of prescriptions of each drug type, metformin and the sulfonylureas, in the US, we see that it's rather stable, the percent allocated to each arm from one year to another. But in the UK, you see the decline here from 2001 onwards for the number of new prescriptions, incident diabetes of sulfonylureas. So, looking at these temporal changes really motivated us to include time as part of the propensity score model. So if we are to look, I know this is, a little bit boring sometimes to look at tables, but it's really important when simulating clinical trials to know what are the differential characteristics of patients, either from one medical center to another or within a medical center between the two treatment arms or among the several treatment arms in the case of antihypertensives. So here I just want to highlight a few points. The patients who initiate on metformin, they're generally younger. They're generally also more hypertensive than the sulfonylureas initiators. They have fewer missing values in terms of both baseline BMI and hemoglobin A1C. So, that can be associated with the year in which they initiated their treatment, but also the physician practice of, recording all of these parameters. In the UK, we observed the same age pattern, and metformin patients were also more likely to have entered the cohort recently and to have lower hemoglobin A1C but higher BMI at baseline. And the reason why this clinically makes sense is that sulfonylureas have a tendency to promote weight gain, and so they are preferable among non-obese patients. So clinically, the distribution of these covariates in each medical center between the two treatment arms makes sense. And actually, the age pattern that we had is very similar to what was found in a prior large-scale analysis that was conducted in 2018. There was a mean age among metformin initiators that was lower. So why looking at all of these statistics and at these tables? Well, one of the assumptions that is needed to identify the average treatment effect is that there should be samples of both sulfonylureas and metformin patients in each group. So that is to ensure that there is some overlap. Given your characteristics, there is a non-zero chance that you receive the drug. So we need to have this positivity. Otherwise, actually, why simulating a clinical trial if in fact you were not eligible given your patient profile to receive a certain drug? So that's the reason why, in addition to these like average values or median values, usually, and this is a step we've conducted, at least for categorical variables, it makes sense to look at all the possible strata and make sure that these strata have, at least one patient of each group. Otherwise, normally this is absence of overlap, and that makes the identification of the ATE more challenging. So the other assumption is that given all of these characteristics, the confounders, we have conditional exchangeability, meaning that now that a weight has been assigned to each patient based on their likelihood of receiving the treatment that they received, we are supposed to have comparable arms where the potential outcomes of the patients would have been the same, irrespective of the treatment they were allocated. So these really are the assumptions that underlie the simulation of the clinical trial in medical records. So visually, we can look at the effect of weighting. And specifically, we can try to determine whether the patient subgroups are more, aligned by looking at the probability density function. And okay, like before any weighting, there might be some differences. So here, for example, we're looking at the distribution of age at baseline, just, visualizing what we were looking at in the table before. The sulfonylurea patients are usually, before weighting, older than the metformin counterparts. However, after weighting, we managed to achieve a much better overlap between the two cohorts. And what is true in the US was also true in the UK. And this is one example with the age variable, but actually we can do that, on average for each of the confounders of interest. And looking at the absolute standardized mean difference, we realize that before and after weighting, there's a considerable difference, and we managed to establish the balance between the two treatment arms. Yeah, it might be beneficial to go faster. Let me know, Alessandro.
Yeah, maybe a couple of minutes. You still have a couple of minutes.
Yeah, so we achieved the same balance in the UK where we had more confounders. So I just wanted to visually show that. And then let me go through some of the results of applying this, two-stage procedure consisting in the propensity score rebalancing as well as outcome modeling with two competing outcomes. So our first outcome of interest, as mentioned earlier, was time to death. So we compared the two drugs on all-cause mortality in both cohorts. So for that, we used a Cox proportional hazard regression model, and we mimicked baseline randomizations with inverse propensity score of treatment weights. As you can see, metformin in blue considerably improved survival compared to the other first-line antidiabetic treatment. And in the US, we didn't find any evidence of a differential effect by age at treatment initiation or by sex. But I just wanted to point out that this is the reason, actually, for investigating, the outcomes of these simulated trials across multiple countries, because in the UK, we found a differential effect based on the cutoff 70 years old at treatment initiation. And we did sensitivity to that cutoff at 65 and 75 and found the same pattern. And in the UK, interestingly, there was also higher benefits with increased BMI at baseline. So these, are interesting facts to consider in terms of determining the populations that might benefit the most from a given treatment. And I just wanted to point out that in both groups, the curves, so the probability of survival curve, they start separating after 3 years, and then they keep like separating consistently over time. So that's also important to look at in terms of like the evolution of the comparative risk over time. So in addition to these hazard ratios, right, which can be, problematic parameters for causal inference, as we discussed earlier, we looked at the probability of having an event by a certain time point t. So we did so in both cohorts for each outcome of interest, so dementia, onset, and death, and for each treatment group. And so what we call the average treatment effect is actually the difference between any two pairs of cumulative incidence functions. And the advantage is that when we do so, we obtain a visual representation of the risk difference, taking one treatment versus the other, with respect to both death without dementia and dementia onset over time. So we could consider risk ratios. That's another metric of interest. But this one really helps measure the comparative effectiveness. And I would like to highlight two observations before switching to the next slide. So first of all, the blue colored curves, the ones that represent dementia onset in each treatment regime, here they were close to each other. And that's the reason why here the risk difference, in the US remains below 0, but in the UK, after year 8, actually crosses the 0. In contrast, for deaths without dementia, the two orange and red curve were further apart, and that's the reason why you see a much more pronounced risk difference here. So we can speak in terms of probability, but I think clinically what matters is talking in terms of months of life without dementia or months of life gain overall. So the risk difference for death without dementia in the full population was clinically meaningful in both data sets, and it was of larger magnitude in the UK, and we estimate that taking the drug for 16 years, if you consider that the patient had years, would bring approximately 15 more months. And the risk difference for dementia onset, were much closer. And there we're talking in terms of like 3 to 4 months of life without dementia over the range of 12 years in the US of follow-up. So these, results are different from one country to another. And so we started investigating, why that could be. So one reason can be the differential distribution of age at baseline. Additionally, when looking at the, incidence of each event, dementia onset or death without dementia, there is a much higher death rate in the UK cohort relative to the US one. So that affects the total number of patients at risk over time. So the total number of patients that are in your simulated clinical trial. And in the US, in contrast, the rates of dementia onset diagnosis, they were much higher. That can be attributed to the fact that we are at a tertiary care center where, neurologists are trained at diagnosing dementia and also at reporting it accordingly in the EHR. So that can explain, why we have over time a different proportion of patients at risk in each, of the two countries, as well as, in each arm. And these population characteristics are important to consider for the design of future trials. So now that we want to partner with a team at Columbia and, design a trial in non-diabetic patients, we really need to take into account the differential rates of mortality, the differential number of patients at risk over time. So that's important for power, but that's also important when making decisions towards inclusion of sociodemographically and geographically distributed groups of patients. So yeah, so these are the results of our sensitivity analysis to your question, Alessandro, earlier about, whether the assumption of a proportional hazard is valid. As you can see, the only problem we had was with deaths without dementia, approximately after year 8 in the UK. However, for the other treatment and cohort combinations, that was fine to make the assumption of a proportional hazard between metformin and sulfonylureas. So that's better highlighted actually when looking at the log of the hazard ratio over time and seeing when there is a divergence with respect to the constant hazard ratio, which is in blue and in red here. So just wanted to highlight that what's interesting going forward is identification, whether manually by stratum or automatically by identifying the clusters of patients that respond the best to the treatment. Here we found that the group of patients initiating on the drug before age 70, performed much better, responded much better to the treatment in terms of both longevity, as mentioned in the UK, and time to dementia onset. So that's really an exciting direction to pursue, automated identification of responders. And that can be really assessed visually by this consistent risk difference below the 0 among patients who initiate early. And as mentioned earlier, whether you consider 70 as your cutoff or 65, 75, that doesn't change your results. And that's another I think exciting direction for people who care about mechanisms of action. And actually, we are doing the same with antihypertensives, where we're trying to assess whether the role of the underlying disease is much larger than the direct action of the drug on the brain. So in the context of diabetes, we looked at the potential role of a better management of diabetes. And in the case of hypertension, I'm doing the same with a better management of systolic and diastolic blood pressure. Although in both cases, it seems like actually the drugs have a much more pronounced direct effect directly on the brain rather than through the management of the underlying disease, which is really interesting biologically. Yeah, go ahead.
So maybe close to conclusion since.
Yeah.
Yeah.
Definitely. So just to conclude, if there was, one aspect to remember from our work, so we found a protective effect on both dementia onset and death without dementia of this anti-diabetic drug. And patients who initiated the treatment earlier seemed to benefit the most from the drug's effect. The results were robust to the choice of the statistical approach, whether a Cox proportional hazard model or a non-parametric model. And it's really interesting to investigate differences owing to population structures. That really helps the design of future trials. And I haven't had a chance to talk about that, but our systems pharmacology experiments in the lab on human neural cells suggested a candidate biomarker, which is a cerebrospinal fluid protein called osteopontin, which could be tracked over time. And it seems like a unique mechanism of neuron protection by metformin is the lowering of levels of osteopontin in the CNS. And so it's, of course, a little bit invasive because it requires lumbar puncture and more than 2 or 3 lumbar puncture per year. That's really the max we can envision, but that could really help track whether that's through the management of this protein's level in the cerebrospinal fluid that the drug acts. So that's actually one biomarker that's being tracked as part of this prospective trial among non-diabetic patients. And of course, as in, all clinical studies, we have limitations, and these, come from residual confounding as well as lack of diversity in the patient population, but also the assumption of adherence to the treatment. If you're interested in these, feel free to email me or look at the paper. And because, a lot of you are in data science and are interested in methodological, developments, I just wanted to mention a few exciting areas to me. So one definitely is better handling of data missingness, leveraging synthetic control strategies or digital twins, as they are called in certain fields, to figure out the trajectory of a patient for whom you don't have much data. Another interesting direction is how to design a big clinical trial in a simulated fashion across several EHR systems. So working with Denmark and Israel collaborators on that. And just to mention another one that I would be happy to have your feedback about maybe during the year in a separate session. Using instrumental variables to me is a good way to alleviate the concern of residual confounding or unmeasured confounding. And so recently I've been deploying these approaches for antihypertensives, antidiabetics, and drugs against arthritis. And I think it also helps us as data scientists understand more of the social processes that underlie all these aging-related diseases. So I found it particularly interesting, both methodologically, because I like econometrics, but in terms of the social science learnings that you gain by doing so. And yeah, thank you all very much for the time, and I would be happy to take additional questions, and feel free to reach out. And I think if you are interested in applying for a master's or a PhD program at IDSS, the applications are open until mid-December. So I just wanted to take a chance also to advertise our institute, because I think it's a unique opportunity to combine statistics and social sciences and have fun for 4 to 5 years.
So let's thank Marie for the great presentation, and maybe we direct questions to emails or something like that for initial time. But really, thanks a lot for a very informative presentation. And I guess whenever you can come to Indiana, you let us know. Okay. Thanks a lot. Thank you very much.
Yeah, for sure. I'd love to come for your regular sessions about data science and social good. Yeah.
Thank you.
Thank you so much.